For today, we're going to be talking about current things going on in biology, and specifically, we're going to be talking about viruses and stem cells.
Okay, so for this next part of class, this is mainly going to be going over topics found in chapters 11 and 12. And really, what I like to do with these chapters is kind of talk about what's presently happening in biology and what's happening in the very near future when it comes to biology. And so since we have this global pandemic going on with the coronavirus, I thought it'd be a nice way to show how does a virus actually work, and specifically, how does a coronavirus work? So I've drawn on the board here our coronavirus, so we can label that. So this is COVID-19. And a virus, the interesting thing about viruses is we call them non-living organisms. Now, if you remember going back to our very first video, our very first chapter, in chapter 1, we talk about life and how biology is a study of life, and how to be considered living, you had to do 3 things, right? You had to be able to evolve, you had to be able to replicate or procreate, create a next generation, and you had to be able to metabolize. Well, viruses can only do 2 of those things. Viruses can evolve and viruses can procreate, but they cannot metabolize. In other words, viruses don't need any type of energy. So one way to kind of think of viruses is they're kind of like leaves in the wind. The environment determines where they go. So there's no type inside the virus, there's no machinery that is helping the virus move any which way or direction. There's no, inside the virus, there's no type of metabolism happening and going on, right? Because you need metabolism for energy. And since the virus isn't using any energy, they don't need it. And so what happens with viruses is they actually, when they infect their host cell, which we're going to see here, they actually use all of the machines of the host cell. They hijack the machines of the host cell to actually replicate. And so that's why viruses don't need any type of metabolism, because they use the metabolism of their host cells. And that's why we call viruses non-living organisms. All right, so once again, here's our COVID-19 virus, and we're going to show you, we have labeled some different parts of it. So the first, we're going to start from outside and go in. So this outside, we have what we call our glycoproteins. Sometimes when you're in the news, they'll refer to these as spike proteins, but this is how the virus is able to attach to its host cell. Then on the next layer is our membrane, all right? So this is what we call our viral envelope. So this membrane structure is the same as the membrane structure of our cell. And we'll see why that's important and actually why that's the case. Then we have our protein coat, which we call the capsid. So this is the protein coat just inside the membrane, kind of gives the structure of the virus. And then on the middle, you have your genome. Now, in our cells, our genome is made up of DNA, right? Our DNA contains all of the genes for our cell and for who we are as humans. Well, in viruses, they can be different types. So some viruses have double-stranded DNA as their genome. Some have single-stranded DNA as their genome. Some have double-stranded RNA as their genome. So remember, we talked about how RNA is mostly single-stranded. This is when RNA, we find it as double-stranded, as it can be the genome of a virus, or it can be a single-stranded RNA for their genome. And specifically for COVID-19, its genome is a single-stranded RNA. So we have here our single-stranded RNA. And that has all the codes on it needs for the spike proteins and this protein capsid. All right, so it doesn't need to be a very long genome, because it's only coding for 2 different types of proteins. So our capsid protein and our glycoproteins are our spike proteins. So the genome is very, very short. And so we're going to show how a virus now will affect our host cell. Now, a virus can only affect the cell that has a complementary receptor to its glycoproteins, and this is how a virus actually infects its host cell. So here we have our host cell. So let's say this is like lung tissue, because that's where we know coronavirus attacks mostly in the human body, is in its lung tissue and causes that pneumonia and syndrome, and what often will go on and cause people to die that can't overcome it. And we're going to draw a small version of our virus. All right, so there's our virus. And what's going to happen is, like I said, it's just moving around in the environment. It doesn't have any metabolism, so it's not like being attracted specifically. But when it enters our system, whether through a cut on our body, or through our nasal passage, or through our mouth, that's how the virus will enter into us. And specifically for the coronavirus, because it infects our lungs, it's when we inhale it that it goes into our lungs, and that's when it can start to attach to the host cells in our lungs. And that's exactly what it's going to do here, is it's going to attach its spike proteins, or these glycoproteins, to the receptors of the host cell. And what that does is it now allows for the virus, and specifically the membrane, our viral envelope, to combine with the membrane of our host cell. This is why it's important to know that the membrane of the virus is the same membrane structure that we see in our host cell. So it combines here with that membrane, and then allows for the virus to enter into our host cell. Once it's in our host cell, the protein coat, redraw that, our protein coat starts to break down. And that allows now our viral RNA to enter into the cell as well. Now remember, we talked about how there's specific enzymes that go around and try to chop up any type of this viral RNA, because they don't want it to overtake the host cell. But if those enzymes don't get to the virus RNA in time, then what happens is this viral RNA will start to hijack the ribosome of the ribosomes of the host cell. And so two things end up happening. Is first, this RNA is going to hijack a RNA polymerase. So remember, RNA polymerase creates more RNA. so that it's going to end up making more copies of the viral RNA. Then another thing it will do, it will hijack a ribosome. And then that ribosome will start to translate the RNA into the different proteins. So what are those different proteins? That's going to be our spike proteins here, so our glycoproteins, and our protein coat, our capsid. Then what will happen is that these will start to come back together to create virus. And ultimately what happens, so our spike proteins start to get embedded into the membrane of the cell. We start to get our capsids coming together and around our new RNA, molecular or viral RNA. And then our virus is going to bud out where these viral areas are all coming together. So eventually you end up having new virus that comes about. And that then is how our virus replicates. So from one virus infecting the cell, you can end up creating hundreds of thousands of viruses that will come out the other side of the cell. And then those viruses will go in and they're floating around and they'll go and infect more cells and so forth and so on. And then that's when you get your disease. We're saying, okay, well, I see that this is how we create the virus, but how does this virus actually make us sick? Well, what ends up happening is, so once this host cell is completely overrun by viruses and this machine making all these new viruses, this host cell will one of two things, either go into apoptosis and kill itself, or eventually the virus will use up all of, the majority of the plasma membrane and the proteins and all that are being created. It will use up everything from the cell, and then that cell is going to die anyways because it can't do its regular work because it pretty much got turned into a virus making factory, and the virus is going to keep making it until it runs out of materials, and then this cell will die. And then as you have multiple cells start dying, that's where you get your disease. So wherever your host cell is, the virus's host cell, is where you see the disease. So once again, because it's coronavirus and it causes severe pneumonia, it's affecting mainly the lung cells of this individual. Now there's other parts where it can affect other parts of the cell. It's also been known that it affects like heart cells and all that kind of stuff as well. But the main symptoms are that pneumonia, that cough, the heavy breathing, all that kind of stuff, because the virus is going in and turning the host cell into this virus making factory, and then it's killing that host cell, and then that affects that lung tissue as more and more of that lung tissue gets infected. All right, so that is COVID-19. That's how you have this problem, this issue going on. Now, one thing you may not know, I don't know if I told you guys this yet, but when I was in college, my main thing that I was studying was viruses. So I'm very interested in everything that's going on with this pandemic, very careful with everything that I do. I suggest all my friends and family to wear masks, to social distance, to everything, because right now, because we don't have a vaccine for COVID-19, that's the only way we can fight against this disease is by stopping it from spreading. And we do that by staying home, or if we go out wearing masks so if we cough and we have it. And the hardest thing is a lot of people are asymptomatic, and so they don't know that they're out spreading it because they don't have symptoms of this. But they're still creating virus because they're infected. It's just not, it's just not hurting them as much as it may some other people. And so they're out there spreading it. Now, when I was in college, my main virus that I was studying was HIV. So that's human immune deficiency virus, right? So I wanted to talk a little bit about HIV as well, because HIV is similar in how it affects the body, but a little bit different. And so we're gonna draw out our HIV virus and kind of show its process as well. Here is our HIV, we just drew out. So our HIV is very similar to our COVID-19. Some obvious differences, so the shape is a little bit different, looks more like a teardrop, where COVID-19 is more circular. Another big difference between HIV is that it has something else in here, and it has this enzyme called reverse transcriptase. And so that's another thing that's different. So COVID-19 doesn't have this, and this is why HIV is very different. It's a very unique type of a virus. We actually call this a retrovirus. I'm going to talk about why that here in a second. But we're going to see that there are similar things that happen. So with our HIV, you've got the glycoproteins, you've got the protein coat, that capsid has your viral DNA. This is also RNA, so single-stranded RNA, similar to the genome here in our COVID-19. But like I said, it has this reverse transcriptase that makes it a retrovirus. And so the same thing is going to do when it infects the cell. It's going to attach to these receptors. So these receptors specifically are called CCR5 receptors. And it's going to enter into the cell. Then the capsid's going to break off, and we're going to see a very similar thing here. We're going to create more RNA. We're going to create our proteins. Now we have another protein that's in there. So our RNA genome for HIV is bigger than the one for our genome for coronavirus, so COVID-19, because it has other proteins. So these reverse transcriptase it has to create. But what happens with these current reverse transcriptase is they are going to do another thing as well. And what they're going to do is they're going to do reverse transcription. So if you remember back to last chapter, transcription was going from a DNA, double-stranded DNA, to a single-stranded RNA to make an RNA transcript. So reverse transcription, we're going to see the opposite happening. And so what's going to happen is these reverse transcriptase are going to be the machines that then turn the single-stranded RNA into that's not a very good picture. Double-stranded DNA, and then that double-stranded DNA actually enters into the genome of the host cell. So what happens is, yes, this virus is going to continue to bud out new virus. So we'll continue to bud out these new viruses. But what it's also done is, let's say one of two things happen. Either our virus, our enzymes inside our cell are able to stop this whole process. So it stops this from going on. Well, what ends up happening is, well, guess what? That viral genome is still inside the nucleus of the host cell, and it could come out and start this process all over again. So either the cell stops the process by using this, or the virus could just do this and hide its genome inside that cell, and then later on turn that cell back into a virus-making machine. And so this is one reason why we have such a really hard time. That's why we call it a retrovirus, because it has this reverse transcriptase and can add its genome into the genome of its host cell. And this is why we have a hard time with HIV, is because it can go latent. Latent means it's quiet, it's not going on anymore, inside its host cell. And so we think, oh yeah, we've killed all the virus out in the environment. Well, then guess what? The host cell gets turned back on and creates more virus. And so it's this really hard thing that we have when it comes to killing HIV and being able to stop it from reproducing. And that's why most of the drugs out there just keep the viral load low for HIV, and therefore makes it so that most people nowadays that have HIV can live a nice, healthy life and don't progress on to AIDS. So the AIDS is the autoimmune insufficiency syndrome that's caused by HIV, because what HIV, the host cell that it's infecting, is the immune cells of the body. So if HIV effectively kills off most of the immune cells, then guess what happens? You become autoimmune. You become autoimmune, so your immune system doesn't work, and therefore a small type of bacteria or virus or something else that usually your immune system would go through and kill, like an easy cold or a flu, would now end up killing you because your immune system has been knocked down so much by HIV. So most people don't die from HIV or AIDS, they actually die from the responses of their immune system now being so low, a small other disease comes in and kills them. So that's one reason why HIV is a little bit more difficult to create vaccines and drugs for, but at least now, like I said, most people that are HIV positive can live a nice long life and never progress to AIDS, because we have drugs that will kill the virus that's out in the open, but ultimately if they stop taking those drugs, the cells will get turned back on and start producing more virus, and then they go to AIDS. And so they have to constantly be taking their antiviral drugs. All right, so one of the cool things about my studies with HIV is I focus mainly on this CCR5. And what has been recently found out in the past 10 or so years is that there are some people that have a mutation in their CCR5 gene. It's called a delta 32 mutation. So delta is just saying a sum of 32. So there's 32 bases that were deleted from the original gene that makes our CCR5 protein. And what that ends up doing is people who have this mutation, their CCR5 proteins end up growing back into the membrane. And what happens is then if they get infected with HIV, the HIV can't actually attach to the host cell, and therefore it doesn't enter to the host cell and never infects the host cell. And then our immune system comes around and cleans up all that HIV. So people who get infected with HIV actually don't get a full infection of HIV because their host cells don't have a good proper CCR5 receptor to allow the HIV to infect their host cells. And so it's found out that roughly 2% of the world population have this. And this is where I did most of my studies when I was in college, is I studied the populations who have this CCR5 mutation, this delta 32 mutation in the CCR5 protein. And I looked at different ways in which we could create drug therapies and maybe even some what we call, not viral therapy. So yeah, I guess you could do viral therapies to give people the gene that gives them this mutated gene so that they would then also be resistant to HIV. This is actually pretty interesting because so they've actually cured somebody from HIV by doing this. And so there was a guy in the late 2000s who had sickle cell anemia. So we talked about sickle cell anemia and how it makes your blood cells, those sickle type things, and therefore you are a higher risk of dying or getting blood clotting or not being able to have enough oxygen sent to the rest of your body. So the normal therapy for sickle cell anemia is to do a bone marrow transplant. So inside your bone marrow, and we're going to talk about this a little bit later when we look at our cells and our stem cells, and there are stem cells that are found inside our bone marrow that create all of our blood cells and our immune cells. And so they all stem from that. So if someone who has sickle cell anemia, so has that bad gene for sickle cell anemia, they can do a bone marrow transplant, and that resets their whole genome for their blood cells and their immune cells, and it gets them back to normal. So this guy who was sickle cell anemic and he also had HIV found a donor who not only had the regular blood gene for hemoglobin, but also this delta 32 mutation. And what happens when they did the bone marrow transplant of that individual, it reset all of his blood cells so they were normal, no longer sickle cell, but then it also reset all of his immune cells to now have this mutation, and therefore the HIV that was in his body no longer could infect other cells, and eventually his immune system cleaned up the rest of these HIV viruses, and now he has been cured from HIV, and he was cured from sickle cell anemia through that one bone marrow transplant that he did. And so a lot of people have been looking at this, or there's other ways we can include this cell or this mutation into our host cells to be able to allow other people who are infected with HIV, who are HIV positive, to also have the same result. So there's been a lot of research in that area. All right, so that is our discussion about viruses. Next, we'll end up talking about our stem cells that I was mentioning. Now we're going to discuss our stem cells. So stem cells are, so if you think back to when we were talking about fertilization, so when we had an egg and a sperm come together to create a zygote, and that zygote then will start to multiply until it eventually becomes a fetus and then a full-born infant, right? So that all starts with the process of these things called stem cells. So stem cells are the original cells of an organism that then eventually differentiate into specific cells. So at a very young age, that zygote is all just the same type of cells. They're stem cells. Then those stem cells will start to differentiate into different parts of the body. Some will be the head, some will be the toes, some will end up being the organs, some would be the brain, some will be the lungs, right? So each of those cells start to differentiate. So what we know is that in each one of our cells, we have the plans for a whole human body, right? In each one of our cells inside the nucleus, the DNA contains the whole plans to create a whole human body. But if you're a liver cell, you only have to have the parts needed for a liver cell opened up in your DNA. So we talked about how we package our DNA, right? So in a liver cell, the DNA is all packaged up except for the part that is needed to run and be a liver cell. And so that's how our stem cells eventually get differentiated into different cells that do different things for that organism. And so that is what a stem cell is. Now, there's 4 different types of stem cells that we're going to be talking about. The first one is our embryonic stem cells, which are the stem cells I was just kind of talking about. So once again, when you first have your sperm and egg come together, they create a zygote. Well, then that zygote is going to split into 2 cells, and then into 4 cells, and then into 8 cells, and then into 16 cells, and so forth and so on until it creates a blastocyst. All right, so this is a blastocyst, and a blastocyst is going to be the early development of your fetus, right? So what happens is all these cells on the outside are eventually going to be turning into the outer parts of the body of that infant. But all the cells that are on the inside, so these ones are all differentiated, because they're going to be the outer side of the body, but these ones are all still regular stem cells. In fact, we call these embryonic stem cells. So these ones in the middle, this top part here, so this part's actually empty. These embryonic stem cells still can turn into any type of cell that we would want them to turn into. And so you're probably thinking, okay, well, why would we want this? Well, think about people who have been paralyzed, right? So they're paralyzed because for some accident or something happened to them where their nerve cells were damaged, and since nerve cells, once they finish developing, they no longer reproduce, so we don't make more and more nerve cells. So if you cut or break or kill a nerve cell, that nerve cell is dead and no longer works, and that's when people get paralyzed. Well, what if you could go get one of these stem cells, create another nerve cell, put it into that person, and therefore now they would be able to walk or be able to move their arms again, or to be able to do whatever it is that they were paralyzed from. So there's a lot of research going on in this area utilizing stem cells for that. Now this could also be, let's say we hear people who have heart conditions and they need to get a new heart replaced. So instead of having to wait for someone to maybe get in a bad accident and then donate their heart, we're able to actually take these stem cells out. And what if we were to take those stem cells and then grow another heart? And we could do this with liver transplants, with kidney transplants. So this is a great opportunity for doing our embryonic stem cells. But the problem was that because we were creating life, and what ends up happening is we will end up killing life once we take out all of these stem cells. And so there was a big ethical issue with this idea that we're going to create life only to kill life just so that we can use these stem cells for medical research. All right, so now we're going to talk about our adult stem cells, which is found in the bone marrow. So these are the stem cells that we talked about before when we looked at the blood cells for people who had sickle cell anemia and the HIV. So this is where those are. So there's these adult stem cells, and they're stem cells specific to blood cells. So blood cells can turn into red blood cells, white blood cells, all the immune cells, your T cells, your killer T cells, your B cells, all that stuff that is used for immune system. So if someone has a disease of the blood, they're able to then, like sickle cell anemia, you could do a bone marrow transplant and give them the blood cells that would then not have that mutated gene for sickle cell anemia, or the mutated gene for the CCR5 for someone who is resistant to HIV and that someone had HIV. So you'd be able to give them that so that they would then become cured of HIV, just like we saw in that example. And so that's our adult stem cells. Now, bone marrow transplants are very painful for the person who's giving them because they can't do anesthesia. If you ever see the movie 7 Pounds with Will Smith, it shows him just like yelling and screaming because he's going through this bone marrow transplant. So another way that's good is doing umbilical cord stem cells. So umbilical cord stem cells are stem cells found in the umbilical cord, but these are also blood stem cells, just like our adult stem cells. And so I have 4 kids, and every time we were having our children, they were asked, do we want to blood bank our umbilical cord stem cells? And blood banking meant just being able to take the blood and saving that later in case we had a situation where we needed to use those blood stem cells. And you'll see this maybe with individuals who have one child that has maybe like leukemia, which is cancer of the blood, and then they will maybe then have another child and they'll take the blood cells from that umbilical cord of that child and be able to do a bone marrow transplant or a transplant of that blood cell with the umbilical cord and help cure that child that had leukemia. And this is really important that it had to be a child that is, a sibling, so closely related, because each one of our cells has these little tags on them that say, I am me, right? So I have all these cells in me that all these tags that say I am Doug cells, just like you have all these cells in you that are who you are. And even though we do things like embryonic stem cells, if we were to create like a heart or something like that, we would still have to make sure that those embryonic stem cells had similar tags to the person that we were going to input that heart, or the body would reject it. And that's where our last one comes into play, and it's very awesome for 2 reasons. First, so these are pluripotent stem cells. So this is like taking a regular cell, maybe like a liver cell, and we talked in our example of liver cells being all packed up, except for just the genes that show for the liver. If you were to unpack all of those genes and revert it back to a stem cell, then you could then give some signals to that stem cell and have it be whatever type of cell you want. So this is called pluripotent cell, reverting back to the regular stem cell state of mind. And this can be extremely beneficial because first it gets rid of the ethical issues of creating life to kill life to then help life. Instead, you're just taking cells from the patient. So let's say it's me and I need a heart transplant, taking my cells, reverting them back to a stem cell and then taking those stem cells and creating a heart cell and giving it back to me. Well, the whole time it still keeps that tag on there that this is a Doug cell, and therefore my body won't reject it. So this kind of kills two birds with one stone of getting rid of the ethical issues and also getting rid of the rejecting of organs and stuff like that issues that you would get as well. And in fact, they've done this already where they took an individual who had esophagus cancer, I think this was in 2017, and they removed his esophagus, and they actually used 3D printing. So remember 3D printers that you've probably used where you input like a picture of a toy, train or something like that, and then it'll print that out. Well, they do that with stem cells where they created an esophagus by 3D printing the esophagus with stem cells that were now esophagus cells. And then they were able to take that esophagus and replace it with the one that has cancer, and now that person was able to live and not have cancer and was cured from cancer by replacing out that esophagus that was created from their own stem cells. So there's a lot of cool areas where stem cells could go here in the future, and we're really excited to see where that's going to go, especially now that we've got rid of those ethical issues that were stopping a lot of the progress that we were seeing with stem cells in the early 2000s.